Hello students, I am Dr. Rajesh Jamudkar, Assistant Professor, Government Medical College, Nagpur. Now we will learn about the transcription process. We will discuss the transcription under the heading of definition of transcription, then various terminologies that are described in the transcription process, steps in the transcription, that is initiation, elongation and termination. We will discuss about the RNA polymerase in eukaryote and in prokaryote, and we will discuss about the inhibitors of the transcription. So what do you mean by transcription process? It is a synthesis of RNA from the DNA molecule. So this is a DNA molecule and genetic information, it is present in a DNA molecule. This genetic information, it is transferred to the RNA. There are three types of RNA, that is mRNA, rRNA and tRNA. These are the three types of RNAs, and this transfer of genetic information from DNA to RNA, it is described as transcription process. The flow of information from the DNA to RNA, and this information it is utilized for the protein synthesis. It is utilized for the protein synthesis. This is known as translation process. This flow of information from DNA to RNA and RNA to protein and its expression, it is known as central dogma of molecular biology. Now, what are the steps in the transcription process? There is initiation, elongation and termination in both prokaryotic and eukaryotic transcription process. There are a number of terminologies they are used in the transcription process, and we'll see that. So first one is the promoter region. Then template strand, coding strand, downstream elements, upstream elements, transcription start site, transcription unit. And we will discuss about the RNA polymerase. So these are the various terminologies that are described in a transcription process. Now this is DNA strand. In replication, entire genome is replicated and both the strand of the DNA act as a template in replication process. While in the transcription process, only the part of gene is transcribed, only a part of gene, and only one strand of the DNA act as a template strand in the transcription process. Okay, so suppose this is transcription start site. It is represented by +1. This 1 indicates the nucleotide. Okay, so before this transcription start site, it is known as, on this side it is known as upstream elements, and after this site it is known as downstream elements. Okay, upstream elements and downstream element. Transcription start site means this is the actual site from which the RNA synthesis occurs. So suppose this is a RNA. This is the actual site from which RNA synthesis occurs. It is known as primary transcript. This RNA is known as primary transcript. Okay. Now there is region which is upstream of this transcription start site. It is known as promoter region. So in prokaryote it is on the coding strand. This is template strand. It is in 3' to 5' direction, and this is coding strand, 5' to 3' direction, coding strand. Now why it is known as coding strand? Because the sequence is exactly similar to that of the newly synthesized RNA molecule. Only difference is that this strand, coding strand, will have the thymine and this will have uracil. This is the only difference. That's why this strand is known as coding strand. And template strand means it is a strand from which RNA is synthesized. It is complementary to the template strand. Newly synthesized RNA, it is complementary to the template strand. Now, upstream of this transcription start site, there is sequence that is known as Tata box. The sequence is TATAAT. It is on the coding strand. It is -10 base pair upstream. This box is known as Tata box, or it is also known as Pribno box. Pribno box or Tata box in prokaryote, -10 base pair upstream of the transcription start site. While there is another region, it is the sequence is TGTTGACA. This is the sequence, and this is region is known as -35 sequence. These are the two region which is before the transcription start site, and this region is known as the promoter region. This is Tata box, it is known as promoter region. What do you mean by promoter region? It is the site, promoter region means it is the site where the RNA polymerase sit on the DNA template. Okay, it is the region on the coding strand. This sequence is on the coding strand of the DNA. So this is the promoter region, this is proximal promoter region. This -35 sequence is the proximal promoter region. So these are the consensus sequences which help in recognition of the promoter region, and it helps in the transcription, starting of the or initiation of the transcription. Okay, so what do you mean by promoter region? It is the region where RNA polymerase can sit. What is the template? It is the strand from which new RNA molecule is synthesized. Coding means the opposite strand of the template strand is known as coding. Downstream elements means before the transcription start site. Downstream element means after the transcription start site, and upstream elements means before the transcription start site. Transcription start site means it is the site from which RNA is synthesized, and this whole unit, it is known as transcription unit, and this one is the transcribed region. This is transcribed region, and this synthesis, the new RNA, it is known as primary transcript. Now this sequence is somewhat different in the eukaryotic. There is this promoter region in eukaryote. It is this is transcription start site. Now -25 upstream, there is one region that is known as TATAAA. This is known as the promoter region. Then -70 upstream, on this coding strand, there is one region that is known as cat box. It is the cat box, and another region is also there, -90 upstream, that is GC box. So these are known as the proximal promoter region. This is known as the promoter region. It is also known as Tata box or Goldberg and Hognes box. This region. This is the site for attachment of RNA polymerase in the eukaryote. Okay. So this is how the promoter region in eukaryote differ from the promoter region in the prokaryote. Now RNA polymerase. RNA polymerase in prokaryote, it is having two alpha unit, then beta prime and omega. This is the core enzyme RNA polymerase. Now one factor is there that is known as sigma factor. Sigma factor. It completes the enzyme and converts this core enzyme into a holoenzyme. So this is RNA polymerase. This alpha, it will recognizes the region of DNA to be transcribed into RNA. Recognizes the DNA to be transcribed into RNA. This beta causes polymerization. Polymerization means 3' 5' phosphodiester linkage formation. Polymerization. Beta prime, it has helicase activity, causes unwinding of the DNA. Sigma factor, it is important one. It scans the prokaryotic DNA, recognizes the promoter region, and help the RNA polymerase to sit on the promoter region. This is the important, and this is the important MCQ, role of sigma factor. Sigma factor scans the DNA, recognizes the promoter region and helps the RNA polymerase to sit on the promoter region. The exact function of omega factor, omega S subunit is not known. Okay, so this is prokaryotic RNA polymerase. Now eukaryotic RNA polymerase, it is of three types. There are three types of eukaryotic RNA polymerase. That is RNA polymerase 1, 2 and 3. Now RNA polymerase 1, it is important in the synthesis of 28S ribosomal RNA, then 18S ribosomal RNA and 5.8S ribosomal RNA in eukaryote. RNA polymerase 2, it is important in the synthesis of mRNA and small nuclear RNAs. While RNA polymerase 3, it is important in the synthesis of tRNA and 5S ribosomal RNA. So these are the three types of RNA polymerase in eukaryote. And there is one important MCQ in the entrance examination. Which one is sensitive to alpha amanitin? It is the toxin from the mushroom. So this is RNA polymerase 2, it is sensitive to alpha amanitin, while the 3 has intermediate sensitivity, while 1 is insensitive to alpha amanitin. So this is MCQ in the entrance examination. So these are the types of RNA polymerase in eukaryote. Prokaryotic RNA polymerase, it is the single RNA polymerase which can synthesize all three types of RNA. Now coming to the steps in the transcription process, there are three important steps in the transcription process, that is initiation, elongation and termination. Initiation, elongation and termination, these are the three important steps in the transcription process. So initiation, elongation and termination. In eukaryote, the process is somewhat complicated as compared to the prokaryote. In prokaryote, the steps are formation of closed complex, means formation of the attachment of RNA polymerase to the promoter region, then opening of the DNA strand, synthesis of the new RNA molecule and promoter clearance. These are the steps in the prokaryotic transcription. But the different factors, various factors are required in the eukaryotic transcription process. Okay, now we'll see the eukaryotic transcription. So this is DNA strand. This is transcription start site, that is +1, and this is -25 region, that is Tata box. Okay, and the -70 region, it is cat box. Now in eukaryote, the special protein that is known as Tata binding protein attaches to the promoter region. This is TBP, Tata binding protein, and it is a part of transcription factor 2D. To this protein there is attachment of transcription factor 2D. Now other transcription factors attached to this complex in a sequence. It is a sequence, that is first is the Tata binding protein, followed by transcription factor 2D, followed by transcription factor A, followed by B, then F with polymerase, RNA polymerase 2 in case of mRNA, then E and transcription factor H. Okay, this is the sequence of attachment of various transcription factor to this promoter region, and that results in the formation of closed complex. After the attachment of this transcription factor to D, it causes the bending of the DNA molecule. That is known as -100 degree bending of the DNA molecule. Now after the bending, this all factor will work in a coordinated manner. This is TBP, this is transcription factor 2D, then the A will attach, then transcription factor to B, then F with polymerase, this is polymerase, then E, transcription factor E, and transcription factor H. So every factor has its own function. This will causes the -100-degree bend in the DNA molecule. A will causes the association of transcription data binding protein and factor to B. B causes the recruitment of F. F brings the polymerase with him. F brings the RNA polymerase with him. E causes the recruitment of H, and H has helicase activity. It has helicase activity. It causes the unwinding of the DNA strand. So that results in the opening of the DNA molecule, and this results in the formation of open complex. This is closed complex, this is open complex. So what are the steps in the initiation? So first one is the attachment of Tata binding protein. Second one is the formation attachment of factors, various initiation factors attached to the promoter region. This is the second step. Third one is the opening of the DNA, local unwinding of the DNA and opening of the DNA. Then fourth one is the initiation of chain synthesis. And the fifth step is the promoter clearance. So what are the sequences? First, the Tata binding protein will attaches to the Tata region, that is promoter region, which brings the transcription factor 2D. It is a part of transcription factor 2D, which causes bending of the DNA molecule. Then other factors will attach to the complex, and then RNA polymerase opens this DNA. After the opening, there is synthesis of nucleotide occurs. Suppose from the transcription start site, this is the +1, and from this side the RNA synthesis will begin. So it is complementary to the DNA strand, and it is in the 5' to 3' direction. It follows the Watson and Crick base pairing rule. So initial chain synthesis will occur. Okay, and after the synthesis of 10 to 20 nucleotide, this RNA polymerase will physically move away from the promoter region, that is known as promoter clearance. Okay, so these are the steps in the initiation process. Now in the elongation, what happens? The nucleotides are added, the ribonucleotides are added, and chain will go continuously. So there is formation of 3', 5' phosphodiester linkage, and synthesis of RNA will continue. Up till it receives some termination signal, the chain synthesis will be continue. It is complementary to the template strand, and there is formation of 3', 5' phosphodiester linkage. But there is no proofreading. The difference in DNA polymerase and RNA polymerase is no proofreading by the RNA polymerase, and second is no primer is required for the RNA polymerase. It can synthesize the fragment at its own, RNA polymerase. So this is the difference. Now how the termination will occur? There is some signals in the newly synthesized RNA molecule. So this is DNA. This is newly synthesized RNA. It is in 5' to 3' direction. There are some signals in the newly synthesized RNA molecule. So it is mainly the A, UAA and CA. These are the signals for the termination of transcription process. This will recruit one factor, that is known as cleavage and polyadenylation specific factor. Cleavage and polyadenylation specific factor. and this will recruit the cleavage stimulation factor. These two factor will recruit the cleavage factor. It has intrinsic endonuclease activity, and this cleavage factor will separate this RNA from the DNA molecule and causes the termination of transcription process. So this is eukaryotic termination and eukaryotic transcription process. Now what is the sequence in prokaryotic transcription? In prokaryotic, initiation is somewhat different. Okay, so there is first sigma factor will scan the DNA, recognizes the promoter region, and it brings the RNA polymerase to the promoter region. This results in the formation of closed complex. Now opening of the strand occur with the help of RNA polymerase, and the open complex formation will occur. Then initiation of chain synthesis. Initial 10 to 20 nucleotides are added, and then promoter clearance will occur. So these are the sequence in the prokaryotic synthesis. Rest of the process is same. Again, there is differ in the termination. In the termination of prokaryote, there is intrinsic termination and row factor dependent termination. These are the two types of termination in prokaryote. In intrinsic termination, in the newly synthesized RNA molecule, suppose this is newly synthesized RNA molecule, this is 5' end, now and this is 3' end. There is GC rich region. So due to this GC rich region, there is hairpin or loop formation. And it is followed by poly U. So it is a intrinsic signal for termination of transcription process. This is intrinsic termination. And another the row factor dependent. This row factor will attaches to the RNA. It goes in a rotating manner, and it has helicase activity. And it will disrupt the DNA RNA association. So this is the row factor dependent termination in prokaryote. So what are the different points in the prokaryotic and eukaryotic replication? This is, first we'll see the prokaryotic, and second is the eukaryotic. So here single RNA polymerase is single, and sigma unit scan the DNA molecule, while here three types of RNA polymerase. And in the mRNA synthesis, the Tata binding protein initially attaches with the promoter region. Then in promoter region, it is -10 base pair and -35 base pair sequences, while here -25 and -70 base pair sequences specific region. So it is known as the Pribno box, while it is known as Tata or Goldberg and Hognes box. It is known as Tata box. There is -35 region in the prokaryote, here -70 region, which is known as cat box. Okay, this is promoter region. This is RNA polymerase. There are three types of RNA polymerase. So very few factors are required here. Initiation factors are required. Factors, that is D, A, B, F, E, H. These are the various factors required for the transcription to proceed. Then in termination, there is row dependent termination and intrinsic termination. While here there is signals, there are intrinsic signals for the termination. So the signals are these signals are, that is A UAA and CA. These are the signals that will stimulate the factors, cleavage factor. Here row and intrinsic termination, in which the loop formation occurs. So this is the different points in prokaryotic and eukaryotic transcription process. Now the prokaryotic RNA don't undergo the post-transcriptional modification. There is no post-transcriptional modifications. Here post-transcriptional modification is there. The primary transcript, it is known as hnRNA, and it undergoes the post-transcriptional modification. Now coming to the inhibitors of transcription process, it includes the first one is the alpha amanitin, which inhibits the eukaryotic RNA polymerase 2. It inhibits the RNA polymerase 2. RNA polymerase 2, it is sensitive to amanitin, alpha amanitin. Then the drugs actinomycin, it intercalates with the DNA and inhibit the transcription process. Actinomycin, it is used in the treatment of certain cancers. Okay, and the second one is the rifampicin. It is a drug which is used in the tuberculosis, and it causes the inhibition of bacterial RNA polymerase. It inhibits the beta subunit of bacterial RNA polymerase and inhibit the transcription process. So rifampicin, actinomycin and alpha amanitin, these are the inhibitors of the transcription process. Now we'll discuss the post-transcriptional modification in the heteronuclear RNA or primary transcript. Okay, so this includes we'll just enumerate the post-transcriptional modifications in the RNA. The first one is the capping, 5' capping. 5' capping, it is with 7-methyl carnosine triphosphate, and unusual 5' triphosphate linkage is there. So this protects the RNA from the nuclease attack, by 5' exonuclease. It is prevent from the exonuclease attack, and it is also important in the initiation of protein synthesis. This cap is particularly important in the initiation of protein synthesis. There is one factor that is known as cap binding factor attaches at this region. Okay, so this is the first. Second is the poly A tail. There are number of 85 to 250 nucleotides, poly A adenine. They are added with the help of enzyme. 85 to 250 adenine nucleotides are added, and it gives the stability to RNA and also prevent it from the attack by the exonuclease. And it is particularly important for the binding of poly A binding protein, which is important in recruitment of 40S ribosomal unit to this mRNA. This is poly A tail. Third one is the splicing. Then this newly synthesized RNA, it will have the cap at the 5' end and various untranslated region that does not code for any amino acid, and this untranslated region, it is known as exon. It is known as introns. So this is known as introns. This is intron, this is exon. Exon means it is a region which is coded for the amino acid, and this is the region which do not code for the amino acid. So it contains introns also, newly synthesized RNA. Now in the splicing process, this intron region, it is removed, and this exon 1 and exon 2 will join together. This is known as splicing. So there are two types of splicing. One is the intrinsic splicing, and second one is the spliciosome mediated. Spliciosome means it is with the help of small nuclear RNAs plus ribonucleoprotein. It is pronounced as SNRP, small nuclear RNAs and ribonucleoprotein. So there are important small nuclear RNAs, that is U1, U2, U4, 5 and U6. They help in the splicing process. So this is known as spliceosome mediated splicing. So here there are steps, that is cut at the 5' end of the intron, then the second step is the lariat formation or loop formation, then cut at the 3' end of the intron and joining up two exons together with the help of spliceosome. So this is known as splicing. Okay, so two diseases they are related with the splicing. One is the SLE, systemic lupus erythematosus. This is the disorder in which autoantibodies or antibodies are produced against these SNRP proteins. Against the SNRP protein. This is one of the etiology in the systemic lupus erythematosus. While because of some faulty alternate splicing, there is thalassemia. Thalassemia is also there because of the faulty splicing. Okay, so this is splicing, and fourth one is the mRNA editing. Editing of mRNA. This is the process by which from one mRNA there are synthesis of two different proteins. Okay, the example is ApoB48 and ApoB100. They are produced from the same RNA, same mRNA, but one is synthesized in the intestine, one is synthesized in the liver. So here the complete production is there in the liver, B100. While because of premature termination, only the protein with polypeptide with 242 kilodalton polypeptide is produced. Because of this cytidine, it is converted into uridylate. Means cytosine is converted into uracil. That's why this CAA, which is code for the glutamine, it is converted into the stop codon UAA, and premature termination is there, and only 48% is synthesized, while here 100% is synthesized in the liver. So this is example of editing. From single RNA there is synthesis of two different polypeptide, ApoB48 and ApoB100. So this is known as mRNA editing. So this is in short discussion of post-transcriptional modification. So this is all about the transcription process in prokaryote and in eukaryote. Thank you.